Okay, folks, let's get started. Hello, class. A couple of announcements. Number one, I forgot to mention last time, if you turned in an exam for regrading, those exams are back in the main office. You can pick those up there. Second, I will be there again for the question and answer session for anybody who wants to come. Last week nobody came. That's fine. If you have questions, come there. And I'll answer. If not, that's fine. I don't take offense. Don't worry. So that's at 4 o'clock today in ALS 4000. I'm sorry, ALS 2018. Oh, I forgot to turn the volume. I wonder why I can't hear myself. Okay, so the last thing I'm going to do here is talk about the very last of the Purine salvage mechanisms. This one. This last one I'm going to talk about, we've sort of glossed over earlier, and now I want to point it out. It's actually an important reaction. It's listed up here above in salvage because it's mostly used in salvage, but it's also a part of the breakdown process for purines. And this involves an enzyme called hypoxanthine guanine phosphoribosyl transferase. It's this mouthful of the name right here. And more commonly, we call it HGPRT. Now, HGPRT catalyzes the reaction that you see here, and it catalyzes this similar reaction with hypoxanthine. Okay, so hypoxanthine is a breakdown product of the purine nucleotides. Okay. Interestingly, this seems to be a relatively minor type of reaction. It doesn't appear to occur in great abundance inside of cells or any of that. However, deficiencies of this enzyme lead to tremendous problems. The medical case associated with it is known as Lesch-Nyhan syndrome. And in patients who have this, they accumulate mostly PRPP. They also accumulate some hypoxanthine. So when this enzyme is not working, those two guys accumulate. The phenotype of people who lack this enzyme is bizarre. Okay, starting at an early age, childhood, the children who have this, who lack this enzyme and who have Lesch-Nyhan syndrome, will start eating their fingers. And it gets worse as they get older, and they actually have to be physically restrained. They will eat their lips off of their mouth. Yeah, it's a very bizarre phenotype, but obviously has neurological implications. It's not known why, as most neurological problems are the case, but it clearly has very devastating consequences for someone who has it. So that enzyme shown right there, it's also listed below on the catabolism, hypoxanthine being listed right here, and Lesch-Nyhan syndrome right there. So we see accumulation of PRPP and also urate, which is, again, a downstream byproduct of purine metabolism. I can't get the words out today. Okay, so that's one of the last of what I want to say about nucleotide metabolism. That actually finishes the last of the metabolism that we'll have in this course. So everything we do from here forward will relate to molecular processes, molecular biology. We'll be covering DNA replication. We'll be covering DNA repair, recombination, transcription, translation, gene expression, immunology, and sensory systems in the next 3 weeks. You think I'm joking? We actually will. So, the first of the topics that we'll be dealing with, of course, is DNA replication. And again, for a lot of you, this is probably going to be some review. I start DNA replication talking about structures. And structure, as you know, is important to understand what's going on. When we think about DNA replication, of course, everybody, as I said, almost every grade school kid now has seen some of this stuff. But the basic strategy of DNA replication is that one molecule of DNA becomes two. And that's essential. If your cell is going to divide, that DNA in the cell has to be completely replicated. And this figure is drawn this way for a very important reason. You'll notice that it starts with 2 blue strands, and it produces 2 products. The 2 products have 1 blue strand and 1 red strand. This means that we now know today, of course, when it was discovered, it wasn't apparent, but we know today that the red strand, of course, is synthesized using information on the blue strand. So the DNA polymerase goes along, wherever it sees a G, it puts in a C, wherever it sees an A, it puts in a T, et cetera, using the rules of base pairing that you know. The result are what are called two daughter strands that have been replicated in what's described as a semi-conservative fashion. So semi-conservative DNA replication excuse me, refers to the fact that replication produces what I just said. Two DNA molecules, each of which has one parental strand and one new strand. When these guys go to replicate, okay, we'll produce four molecules. And of those four molecules, from the starting molecule, there's only two strands that are present. So only two would even actually have that. But the two new strands that are made would have one of this and one of this, and then one of this and one of this, and then new strands going associated with each one of those. So that's what semi-conservative replication is. That method of replication was actually discovered before replication itself was even understood. It was discovered by two professors known as Messelson and Stahl. Very famous experiment that they did that I'm not going to talk to you about here. But Frank Stahl, later in his career, was a professor at the University of Oregon. And they won the Nobel Prize for their discovery of that structure of the replication of DNA. Okay, now, we know today that replication, in addition to replication, DNA has to undergo other processes. One of the things that happens to DNA molecules, because we are so prone to getting all these other things in our diet, in our environment, and in our air, and in our water, is damage. That damage can be chemical. That damage can be radiative in nature. That damage can be in other forms as well, adducts added on to DNA and so forth. So that means that cells really need to have mechanisms to repair that damage. And I'll spend some time talking about different repair mechanisms after I talk about DNA replication to elucidate some of those mechanisms that they use to keep from causing problems. If cells don't repair their DNA, if your cells didn't repair their DNA, you'd be dead today. Okay? One trip out in the sun and you would be dead. There's enough mutation going on just by exposure to sunlight that if you don't fix that and don't repair that, it would lead to cancer and death at a very early age. Okay? So these repair mechanisms are very, very important for us to understand. All right. Well, as I said, I like to start this discussion talking about structures of DNA molecules. DNA, the structure that we think of as the structure of DNA today, is the most common structure of DNA. It's referred to as the B form of DNA. And it's the structure that was determined by Watson and Crick back in 1953. They determined that structure by stealing data from a woman named Rosalind Franklin in a story that's become very famous. And they essentially acknowledged they stole the data. But they got the credit. She didn't get the credit, even though she was the only person in the world at that time who knew how to diffract X-rays through DNA. And a pretty remarkable woman. The structure that they described is called the B form of DNA, and we know it today as occupying probably about 99% of the structure of the DNA in a cell. So DNAs in a cell are not all in the B form, of course, but a pretty good percentage of the DNA is in the B form. Rosalind Franklin was, at the time, working on a related structure of DNA that a lot of people don't realize she published in the same issue of Nature that Watson and Crick published the B form in. She published in the same issue of Nature the structure of the A form of DNA. The A form is a rarer form of DNA. And when it was originally published, it was felt that it probably didn't occur in cells at all. The reason that was felt was because the way that she made the crystals to study, or the fibers to study this structure, was that they could only form under her conditions if she had absolute dryness. So she had to dry her samples out extremely in order to make these structures. And it was felt that, well, DNA is in an aqueous environment. It's unlikely we're ever going to have DNA in the cell in this form. So therefore, the A form is probably some sort of an artifact that's there. As I said, we now know today that the A form of DNA does, in fact, form inside of cells. And one of the things that the A form of DNA is associated with is bends in the DNA. Okay? it has a sort of a bend associated with it. Now, there are many things that can bend DNA, and we'll talk about some of those later, but having an actual thing in the structure that causes a bend probably is very, very useful. The example I like to give, and I've probably given it in this class before, if not, I'll mention it again, is that DNA is a very long molecule. If we look at the DNA in our cells, all the DNA in one cell, 7 feet. So we got 7 feet of DNA that's got to be coiled up and wrapped up. And it is coiled up and wrapped up, but having nice adjacent bends with which to facilitate that bending, that wrapping up, is probably a good thing. So the A form of DNA turns out not to be a trivial form of DNA. In the late 1970s, a professor at MIT known as Alex Rich got studying a DNA molecule at a much higher resolution than was able to be done in the 1950s. And he discovered a structure of DNA that when he first published it, people said the same thing they said about Rosalind Franklin's. Well, it's clearly an artifact. And it turned out not to be an artifact, and I'll tell you a story about that in just a second. But the form of DNA that he discovered, we refer to today as zDNA. Z standing for zigzag. Because in contrast to the A form and the B form, which make a nice, smooth helix, the Z form of DNA is stretched out. So the bases are further apart from each other. And when you look at it, it looks like it goes in a zigzag structure. Another aspect of the Z form of DNA that I will describe to you, and hopefully it'll make some sense when I describe it later, is that the strands are twisted around each other in the opposite way that the strands are twisted around the A form and the B form. So zDNA has strands twisted in what we refer to as the left-handed form. They create what's called a left-handed helix. Now, you probably never thought about it before, but it turns out that there's two different ways of wrapping strands. Okay? If you like to think of them looking from above as clockwise or counterclockwise, that would be appropriate. But it turns out that the two forms are referred to as left-handed and right-handed. So A form and B form are both what are called right-handed helices. And the Z form is left-handed. And because it was left-handed, people said, oh, well, no way that's going to form in the cell. Because if I've got a DNA molecule, okay, I know that the vast majority of the DNA in the cell is in the B form, maybe even the A form. How in the world can it twist this way and then untwist back the other way and then twist again? There's no way that's going to happen. Okay? This would create something that looks very, very unusual. It would be very difficult for it to flip. Well, never say never in science. People got studying the Z form of DNA very carefully, and they were able to demonstrate that, in fact, the Z form of DNA does form inside of cells. Okay? Now, I'm going to tell you this, that's where one of my stories comes up today. I'm going to tell you a story about that. So the story I have for you is that we had a professor in our department who was the chairman of our department for several years named Dr. Xing Ho. And Dr. Ho had been a postdoctoral fellow with Alex Rich at MIT. And so he was quite familiar with the different forms of DNA and the crystallographic techniques that were used to study those and so forth. And so when he arrived at OSU, he began studying the Z form of DNA, among other things, that he was interested in. And Dr. Ho was very interested in the sequences that might give rise to Z. Because we know that the sequence of DNA varies. We might see slightly different structures in some form as a result of those sequence differences. So he started analyzing, on a crystallographic basis, the different tendencies of different DNAs with different sequences to form, either B or Z. And what he found was that there was, in fact, a sequence dependence. Okay? So he could actually write a computer program to predict, based on sequence, the likelihood that a given stretch of DNA would be in either the B form, or the A form, or the Z form. This is pretty cool. Pretty darn cool. Okay? So that was a very interesting and cool finding. So the story picks up then, a couple years later. This program that he had written, which was called Z-Hunt, was useful. You could upload a sequence of 10,000 base pairs to the website, and it would come back and it would say, oh, the likelihood this is going to be in this sequence, stretch is going to be Z and B and so forth. You get a plot, a probability plot of the likelihood of being in these various structures. So at about that time, I had a student, he had an undergraduate student who was a pretty good computer programmer who wanted to work in the HHMI program that I direct. And they wrote a proposal saying that they wanted to take this Z-Hunt program and put it to work on a much bigger problem. Instead of these little sequences that people were sending them, let's take a look at a human chromosome. And that was about 2001 when the first entire sequence of a human chromosome was published. And so Christoph, that was his name, Christoph, took this Z-Hunt program and rewrote it so that it could handle these bigger sequences that were present in human chromosome 22, which has several million base pairs, way bigger than anything that had been analyzed before. So he ran this analysis through the program, and when he finished it, we all realized he'd done something very special. He was the first person on Earth ever to completely characterize the structure of a human chromosome. He was a sophomore in college. Okay? Now, pretty cool. He got a first author publication out of that, which itself was pretty darn neat. Okay? But then, when they started analyzing the data, they discovered something even cooler. So he said, okay, well, here's the likelihood of stretch of this. So you know all the places in human chromosome 22 where Z form actually forms, or the likelihood that it's going to form there, the most likely places it's going to form. And so they asked the question, well, are these interesting parts of the chromosome? Okay. And it turned out they were. When we look at eukaryotic chromosomes, I'll talk about them later, but we look at eukaryotic chromosomes, what we discover is that eukaryotic chromosomes have a lot of what we call junk. It's not really junk, but they're long regions and long stretches that don't code for any proteins. So in eukaryotic chromosomes, there's big gaps between the places where the actual proteins are. That's different from bacteria. Bacteria have the genes right next to each other. Very tight, very compact. Eukaryotic chromosomes, you go for a long ways, no DNA, no gene. Okay? Well, interestingly, they found a relationship between the place where Z forms and where genes are. Now that, I think, was even more magical, because now you started to have an idea about why Z was important in the first place, because it's probably a little flag to the cell saying, hey, there's a gene here. There's a gene here. And so in this long, long chromosome, there are markers telling the cell where to look for genes. And it's probably in the form of zDNA, at least to some extent. So that was a pretty cool finding. And I always like to tell that story because, A, it does tell the relationship between structure and function. The structure of DNA giving rise to an important function, which is telling where the genes are. But I think it also tells about what's possible to do as an undergraduate. And that's why I like to encourage undergraduates to get out and do research, even if you don't do it in my program. Okay. So, Z form is an interesting and different form of DNA. When we look at the structure of DNA, there are two major features that we describe with those structures. One is known as the major groove, and the other is known as the minor groove. And these are shown fairly clearly on this figure. The major groove corresponds to this big gap, and if we were to do it in 3 dimensions, we would see this gap wrapping all the way around this thing. The minor groove is this little tiny thing here. So if we were to look at and describe the structure as we went from bottom to top, we would see minor groove, major groove, minor groove, major groove, minor groove, major groove, alternating through there. And this structure is present in all of the 3 helices that we see, even the left-handed Z form that is a little backwards. Now they vary from each other with each other in how big the major and minor grooves are, and we're not going to worry about statistics or numbers of that. Okay? This is a figure that I'm not going to talk about here, but I had a student ask a question about earlier. The major groove and minor grooves arise, and they can be visualized here. And if you don't picture this, don't worry about it. We're not going to draw it on an exam, and we're not going to hold up a model and have you tell me which is major and minor. But the major and minor grooves do have distinct features that are on each side of them that give rise to hydrogen bonds. Okay? So we can think of that minor groove and major groove as things on the outside of that molecule. And what they're pointing out are different hydrogen bonds. They're different than the hydrogen bonds that we see in here. Well, actually, these two are in fact not. In other cases, there are like a nitrogen here that can affect things and so forth, a nitrogen there that can affect things. that these other things that can give rise to hydrogen bonds on the outside of the groove allow proteins to, quote, read them. So a protein that binds to a specific sequence doesn't have to get in the middle of this thing to figure out where it's at. It has to read either on the major groove where the hydrogen bonds are, or on the minor groove. And the base pairs have different sets of hydrogen bonds that they're throwing out. So the bottom line is that the composition of the base pair can be read by the hydrogen bonds that are in the grooves. If you're in the groove, man, you're there. Okay? That makes sense? All right, so that's kind of cool. Now, we'll talk about later about proteins that bind to DNA. Most commonly, those proteins are called transcription factors if they bind specific sequences, and we'll say something about those later when we talk about transcription. Okay, here's a table that you'll ignore and I will ignore, but I just use it to show you just a couple different things about the different forms. Here's the A form, the B form, and the Z form. Okay? The narrowest is the Z. Remember I told you how it's stretched out? Not surprisingly, when you stretch it out, you stretch out a piece of spaghetti, it's going to get narrower. And that's what happens here. It's the narrowest. B form is intermediate. And A form is the broadest. And what that means is that A form is essentially scrunched. So A is the most scrunched. Z is the most pulled out. Okay? Base pairs per turn of the helix. This is something that we'll be talking about in just a few minutes that's important. Base pairs per turn, and we're going to use this number to make it simple. We're going to call this 10.5 and not 10.4. We're not going to, the number itself isn't that important, but 10.5 is what we're going to use for calculations. The number of base pairs per turn means the following. If I think about a DNA molecule, if I start here and I go up the helix one complete time around, the question is how many base pairs are in that one complete revolution? Okay? And for the B form of DNA, that complete structure, or that complete turn takes 10.5 base pairs. 10.5 base pairs per turn. When DNA is in the B form and it has 10.5 base pairs per turn, we're going to see some things can alter that. But when it's in the B form and it has 10.5 base pairs per turn, it's what we describe as relaxed. It's relaxed. It's had a beer, okay? It's chilling, it's relaxed. On the other hand, if we start twisting the strands, we can make it go to a different number of base pairs per turn. And just like what happens to you during exam time, it's not going to be relaxed anymore. It's going to be in a very tight and taut state. And just like you are at exam time, it's going to do whatever it can to relieve that tension. And that comes back to the beer thing again. Okay. All right. So that's the basic things about structure. Now, I need to introduce a topic to you that most students find more confusing than I think they should. And maybe it's because I'm not very good at explaining it, which is entirely possible. But I will try. Okay. If we think about a DNA molecule, we can think about a DNA molecule existing in two forms. We can think about it existing in a linear form, as we see here. And if I were to take and put these two ends, join this end over here to this end over here, I would make a circle. Okay? Now these are fundamentally different things. They're fundamentally different things. For example, if I have this guy right here, and I have a fairly short DNA, and I grab one end of this thing and I start saying, okay, I'm going to untwist this guy a little bit. What's going to happen to this thing? Well, the reason, remember I said when we're sitting around in the relaxed state, when it's relaxed, this is the state it wants to be in. If I lay that DNA molecule out on this table right here, it's going to lay there just like this, okay? 10.5 base pairs per turn. It's as happy as it can be, just laying there. If something comes along and tries to twist it tighter, it's going to resist it. And if something tries to loosen it, it's going to resist it. Make sense? That makes sense, right? Well, if this is linear, if I start twisting this end, that gets communicated all the way through over here, and it untwists over here to make up for it. Or if I start loosening it over here, then over here it's going to start tensing up to replace what I loosen it with. So any changes that I try to make to this linear molecule are going to be resisted by the fact that it really wants to be at 10.5 base pairs per turn. Okay? That's built into its structure. It wants to be at 10.5 base pairs per turn. All right, now that's our starting point. Let's imagine then I take, as I said, those two ends and I join them together. And when I join them together, I make a circle. Okay? Now we see some terms on here. We'll not worry about the terms for the moment. I've made a circle. And in fact, what I have made is I've made a relaxed circle. Because I didn't do anything. I didn't twist it. I didn't untwist it. I just grabbed the ends and I stuck them together. Everybody with me? That's all I did. I just stuck the ends together. All right? So that circle is going to sit there and it's going to be relaxed. Now, imagine if I had taken that circle and before I put the ends together, I said, oh, I'm going to hold on to this end so it can't untwist. And I'm going to take this end and I'm going to go dring, 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 twist it, right? Then I join the two together. Now, it can't come apart. The other end can't relieve the tension that I would have put into it if it were linear. This is why the two forms are very fundamentally different and fundamentally important. Okay? What do you suppose that DNA molecule is going to do when I do that? Well, it's going to be all it's going to all wrinkle up, and I'll show you why it's doing that in a second. So you imagine a rubber band. You take a part of it, and you just keep winding, winding, winding. Okay, it's going to get all coiled up. That coiling we refer to as supercoiling. And I'm going to show you a picture that demonstrates this. Okay, this is an electron micrograph of two different DNAs. This guy on the right is a relaxed circle. The guy on the left is exactly the same size. It's been supercoiled. Now, we see that the cell has an advantage in the first place to making a DNA be supercoiled because A, it can occupy less space. Right? So the cell had taken it, put in, or took out. Either way, it doesn't matter. It doesn't matter in the sense that you can make a supercoil by putting in extra twists or taking out twists. They're called negative and positive supercoiling. It doesn't really matter any. It doesn't matter. They both will give a structure that looks like this. Okay? So supercoiling has a biological function, and now we need to understand how and why that happens. Okay? Any questions about this before I start explaining the calculations? Okay, so the second benefit of this is that what we will see, and it's kind of, you don't see it on this figure because the resolution isn't good enough, but what happens is this tension can cause the strands in some places to come apart. And this can affect how well transcription occurs. We'll talk about it more when we talk about transcription. But that's something just to keep in your mind, that supercoiling affects how well the strands stay together. Okay. Now, let's get into some calculations. All right. So here's my molecule. Now I'm going to do what I said. I'm going to hold on to this end, so this end can't relieve any tension I take out here. I'm going to unwind this guy a couple times. And now I'm going to put the two pieces back together. Before I do that, I need to define something for you. Okay? Twists are pretty easy to understand. Twists are the number of times these guys cross over each other in this strand. This guy started out with 25 twists. I've just unwound it twice, so now it's got 23 twists. Okay? Do you suppose this guy is going to be under a little tension? Right? What happens if I let go at the other end? The tension will be relieved, and I'll have 25, right? Makes sense? If I were to twist it 2 more times instead of untwisting it 2 times, I would also have tension. And when I let go of that end, it would relieve that tension. And I'd be back at 25 again. So we can see that the DNA molecule wants to have 25, this particular one wants to have 25 turns. Because 25 turns give it 10.5 base pairs per turn. When I alter the number of turns, and by the way, I use turns and twists interchangeably. When I alter the number of turns, okay, and I have the same number of base pairs, then that ratio of 10.5 base pairs per turn is going to change. Right? Everybody with me? That's what the tension is. The tension is anytime I get away from 10.5 base pairs per turn, this guy wants to flip back into it. So there's an energy associated with this. An energy. Okay? And that energy is realized if we let go of the other end, because boing, we see the untwisting or the retwisting occurring to get back to 25 twists, which corresponds to 10.5 base pairs per turn. Okay. Now, what if I don't let go of the other end? What if I join the ends like I said? So let's do that. Let's join the ends and make a circle. Well, now the other end can't go floating freely. One of the ways this guy could exist would be to look like this sort of big mouth thing up here, this big loop. But remember, this molecule is under tension. The molecule is under tension. So that molecule would like to relieve that tension, and the molecule would love to get back to 25 turns. Right? And so it does. It's that desire to get back to 10.5 base pairs per turn, in this case 25 turns for this molecule, that get us the supercoiling that occurs. So what will happen is the following. Notice that I have 23 turns in this molecule. This is the same molecule. It has now done a little trick. The reason it coils up is the coiling up allows it to twist itself more. So the twists, it retwists itself. Okay? It can't take the ends apart. If it could take the ends apart, then it would just go to 25. So to relieve the tension of getting to 25, this thing loops around itself. It forms what we call supercoil or superhelical turns. Now, we're back at 25. If you count them, you'll see 25 of these going around there. But because the ends were closed, we couldn't just unspring it. The springing happened internally. And that's why we get these two superhelical turns. That's why that molecule that you saw in the electron micrograph is so scrunched up. Because internally, this guy, that molecule was getting back to 10.5 base pairs per turn. And the only way it could do it was by putting superhelical turns in there. The superhelical turns are a thing we call writhe. W-R-I-T-H-E. A DNA molecule writhes in order to allow twisting to occur to get back to 10.5 base pairs per turn. A DNA molecule will writhe to allow twisting, or in some cases untwisting, to occur to get back to 10.5 base pairs per turn. Now, you understand what twists are. In order to make this, to put in those two extra twists, I had to, in other words, in order to put in the two twists in the strands, I had to essentially untwist the bigger molecule. So I put in two additional turns here. I take out two turns of the molecule, and that's a negative writhe. It's a writhe of a -2. All right. Now, how do I know if it's negative or positive? We'll come to that. Okay? The last term I need to describe to you is the linking number. The linking number is equal to the number of twists within the strand, which is 25, plus the number of writhes, which in this case is -2. So the linking number is equal to the number of twists plus the number of writhes. Okay, now let's do a little thought exercise on this. We're going to try to avoid calculation. We're going to think this through, okay? I've got a DNA molecule. It has 12 base pairs per turn, and I put it into a circle. What's it going to do? Is it going to writhe positively? Is it going to writhe negatively? And why? It's got too many base pairs per turn, which suggests it has not enough turns. I have unwound it, right? Right? Not enough turns. So I have unwound it, because if I've reduced the number of turns, I have unwound it. If I have unwound it, what do I need to do to get the thing to be more relaxed? I've got to put more turns into it. Right? Everybody with me? No. I've got to put more turns into it. I put more turns into it. I've got to therefore counteract that with the same number of writhes. So the writhe will be a negative number. The turns will be whatever it took to get me back to 10.5 base pairs per turn. Is the linking number going to be greater than or less than the twist? It's going to be less, because twist plus the writhe in this case is negative, will give rise to the linking number. All right. Now, I know that's going to be a little confusing to you, but there are some problems in the book I'd like you to look at, and I'd like you to think about. Okay? And if you still find it confusing, please come see me, and I'll be happy to step you through these and explain them to you. I will take a couple quick questions on that right now, though. Any quick questions I can tell you? You want to think about it. Okay. All right. And let's see. Okay, so what I've been describing to you are different forms of the same DNA. In other words, I didn't change the sequence. I didn't change the composition of the DNA. All I did was I changed the number of times I twisted the thing. Right? So we give a name to different forms of the same DNA. The name that we give is we refer to them as topoisomers. They are isomers of each other. And topo refers to topological. So they have a different form. You saw that supercoiled circle was different than the relaxed circle was. They're topoisomers. Now, it turns out that cells have enzymes that can make different topoisomers. Enzymes that can actually change the linking number of a DNA molecule. Okay? So these enzymes are called topoisomerases, Topoisomerases, if they're plural, topoisomerases. And there are many different ones that cells have. I'm going to describe a couple of them to you right here. Okay? What you see pictured on the screen is a bacterial enzyme known as topoisomerase 1. Now, topoisomerases are grouped into two specific categories. The two categories are they're referred to as either type 1 topoisomerases or type 2 topoisomerases. The type 1 topoisomerases work by cleaving a single strand. The type 2 topoisomerases work by cleaving both strands of a DNA. Now, I'll explain how they work in a second, but that's the primary difference. A type 1 topoisomerase works by clipping one of the strands. And so what it does, it clips it, it lets it do its thing, and then it puts it back together. A type 2 topoisomerase clips both strands and then puts both strands back together. Why is it doing this? Well, let's think about this. Let's imagine I'm a DNA polymerase running down replicating DNA. When you're replicating DNA, everybody's seen the replication fork, right? You got the two strands, right? You got the DNA polymerase scooting along there. What you probably never thought about was that these strands are being pulled apart as you're going along. If you're pulling the strands apart here, what's that going to create ahead of where you're pulling the strands apart? It's going to create a tangle. It's going to create a topoisomeric mess. Because you're changing the twists ahead of it, right? If you don't relieve that tension, that DNA is going to make a knot, and replication is going to stop. Well, cells use topoisomerases. There's several things they use them for, but one of the things they use them for is to keep those knots from forming during replication. Now, I'm going to tell you about topoisomerase 1 first. It's not a primary one for relieving tension, but that's the way I'm going to describe it. Let's imagine I had that circular DNA I had before. And you saw that looped out region, right? Before it supercoiled, you saw that looped out region. It was under tension. That tension is building up. It's building, it's building, it's building. What if I want to change that tension? Well, I bring in a topoisomerase 1, and what a topoisomerase 1 will do is it'll grab onto the DNA. There's the DNA. There's the topoisomerase. And it will clip one strand. And when it does that, all of a sudden, all that tension that was in there, remember before when we had both strands, we could just let it relax? Now that one strand that's loose will go zzz, unwinding, relieving that tension. Okay? So a type 1 topoisomerase is clipping one strand and allowing the strand that it clips to just unwind, 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 relax. A type 1 topoisomerase can also put tension in, can do the opposite thing. Okay? But it's doing everything by clipping one strand and leaving the other strand intact. That's very useful. And the reason that it's useful is once you're done, everything is held in place. All you have to do is join those two ends that got clipped in the first place, and then you leave and everything is hunky-dory. So type 1 topoisomerases have it relatively easy. The type 2 topoisomerases have a more difficult challenge. And they do it, but it's in your head harder to get a handle on. Now, in the case of the type 1, since the DNA double helix is half together, that one strand is still holding everything together, like I said, all we have to do is bring these two pieces, the two other ends together, and then join them, and we're done. So the action of a topoisomerase requires clipping, doing something about the tension, and then rejoining those strands. The type 2 topoisomerase cuts both strands. Now, if nothing else were to happen, we would say, whoa, this is a cool problem. This guy's going to unwind, 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 unwind. But what holds everything together? This is a bigger problem. And it turns out that the type 2 topoisomerase has to hang on to both ends. And the way it works is by passing a strand through itself. Now, the mechanism you won't understand from that brief explanation I just gave you, but trust me, it passes a strand through itself. I'll show you a figure in a second. Okay? Type 2 topoisomerases are the ones that are actually most involved in E. coli replication at relieving tension. There's a type 2 topoisomerase known as gyrase, DNA gyrase, G-Y-R-A-S-E. DNA gyrase is a type 2 topoisomerase, and the place you find it is ahead of the replication fork where all that tension is building up. And that guy is handling that tension and keeping the DNA from turning into a knot. Pretty cool stuff. Okay, now, the important thing about, now I was going to show you a type 2 topoisomerase here. Let me do that for you. So here's a type 2 topoisomerase, and its mechanism is, as I said, it actually passes a strand through itself. It opens, passes through, and does its thing. Type 2 topoisomerases require ATP. Something's got to be managing all this thing and all this movement, and they require ATP. Type 1 topoisomerases do not require ATP. At least not all of them do. Okay. Now, topoisomerases, as you might imagine, are pretty essential for a bacterium to do its thing. Because if you screw up the topoisomerases, what's going to happen during DNA replication? You're going to form a knot. Okay? Replication is going to stop. If replication stops, the bacterium can't divide, and you've just killed the bacterium. So not surprisingly, bacterial topoisomerases are targets of drugs. Bacterial topoisomerases are different enough from our topoisomerases that in some cases we can make a drug that will completely affect them, that will not affect us. Ryan, you had a question?
Do they just take jumps in the DNA and cut it when they want to jump in when they want, or is this like continuous?
So, good question. So Ryan's question is, how does the sort of mechanism by which the topoisomerase does this thing? Does it jump? Does it move along? And it's sliding along the DNA. So we'll see when we talk about the replication of DNA that there's a giant complex that literally is sliding along DNA. And many DNA binding proteins that are not specific for sequence, that is, they don't bind to a specific sequence, but they bind to DNA in general, like a topoisomerase, will use that sliding mechanism. They just slide right down the strand. Laurie?
Do you have type 1 and type 2 working in concert, or do you have one or the other?
Yeah, it's a good question. So do type 1s and type 2s work in concert, or one versus the other? You tend to have one versus the other. Some are specialized for different things. I'll give you a brief story here since you've asked. The gyrase is almost exclusively the one that's evolved ahead of the replication fork, relieving that tension. The type 1s get involved in other topological concerns that we'll talk about with respect to transcription. Okay? But as I said, there are many different topoisomerases. There's a very cool one that's a type 2 topoisomerase that bacteria have. It's called Topo7. Okay? And Topo7 has a very simple function. Very, very simple function. And it's a function that we don't have in our cells. Our chromosomes are linear. Bacterial chromosomes are circular. So when you replicate a bacterial circle, you start out with one circle. At the end, you have two circles, and they're intertwined. They're intertwined. Bacteria have Topo7 that does one primary function, and that is to take apart, pass through, let out. So the cell division can occur. And now you start thinking, wow, antibiotic, right? Here's something that we don't even do. If we knock this guy out, we stop bacteria from dividing, we kill them, and in fact, Topo7 is a target for antibiotics. Okay? It only does one thing. It allows those two chromosomes to separate, but it's an essential function for bacteria. So when you're designing an antibiotic, having creativity, thinking about how we might target something different from what bacteria have from what we do, can be a very, very useful thing. And that actually has happened in the case of topoisomerases. Okay, well, I'm going to finish today by talking about some specific antibiotics. So there's an antibiotic that's not pictured here. It's called novobiocin. And in fact, all the antibiotics I'm showing you here all work on DNA gyrase. They all work on DNA gyrase. So DNA gyrase is pretty important. If you can't, like as I said, if the cell can't unwind it or relieve that tension, then DNA replication is going to stop. Novobiocin prevents the DNA gyrase from being able to work with ATP. It prevents it from using ATP. So if it can't use ATP and ATP is required, the gyrase is dead, the cell is dead. Okay? Nalidixic acid, and again, you don't have to worry about the structures, but the names I think are important. Nalidixic acid and Cipro. You guys have heard of the antibiotic called ciprofloxacin. It's a relatively new antibiotic. Ciprofloxacin both work on DNA gyrase, and they inhibit the ability of the gyrase to interact with the DNA itself. So since it can't associate with DNA, it can't go in and do its thing, and therefore the bacterium is dead. Cipro has been used increasingly with different, now that there are different various strains of bacteria that are resistant to other antibiotics, and this has been very effective in treating some of the drug-resistant bacteria that aren't yet quite resistant to ciprofloxacin. Okay, that's a good stopping point for today. I will see you guys, if not at the questioning session at 4 today, I will see you on Friday.
Last term we were talking about plasmids and do you have a question?
Second.
How do you prevent these things from like running into each other? They got multiple things running around on the DNA.
It's a pretty good question. You'd be surprised. It'll actually get more complicated when I talk about transcription. I don't have an answer for it, but there's some surprising things that can happen where there actually is traffic that seems like it's going to collide and it just goes smoothly across each other. It's not really known. Okay. The problem is that these things happen very fast, and so it's like trying to, study a car crash with a camera, and like a single camera, you can take one picture. That's really what you get. So, okay? Yes?
How does a topoisomerase know that there's a change in tension of the molecule?
So, yeah, in fact, what happens is that the DNA itself will change in structure, and those structural changes are communicated inside of the topoisomerase. Okay?